So I would like to start by thanking the Cystinosis Research Foundation, because I have to say that none of this work would have been possible without the foundation. At the beginning of this project, nobody believed in it, and nobody would have bet a penny on it. And so the foundation really gave me my chance and supported all the project. And so now we have a government that found also part of this project, but before they wouldn't have done that, before they see that it was working. So thank you, the foundation, and thank you, everybody, to be here. So I will talk about the hematopoietic stem cell transplantation and gene therapy for cystinosis. I will try to explain what is hematopoietic stem cells. I mean, I will do a less good job than Betty yesterday, but I will try my way. So the hematopoietic stem cells are, they are stem cells that reside in the bone marrow. So everybody has stem cells in their bone marrow. And they stay here all your life. But if you have any injury anywhere in any organs, they will be called and go and mobilized in your blood and go to the targeted tissue to try to repair it. So that's their function. I don't know if you see what I just said. Because there are several kinds of stem cells, and I will just talk about the hematopoietic stem cells. So these cells, they are safe because we can use them in clinic for some kind of cancer of blood every day. So they can be transplanted. They are transplanted for different reasons every day. So we can use them. You might hear later on that the stem cell in human are called the CD34 positive stem cells, and we can isolate them easily in your blood. So we can just do a blood draw and take this stem cell from your blood. So what did we do? We used the mouse because, as we said yesterday, the problem with cystinosis is that the cytinase gene is expressed in every single cell of your body. So this means that if you want to do a gene therapy approach, you have to bring the gene to every single tissue. That's why at the beginning I was thinking the only way to do that is that you need a vehicle to do that, right? And that's why I thought about the stem cells, because theoretically they are supposed to be able to go to any tissue if there is injury. So that's the approach I was thinking at the beginning. But of course we need to prove that first in an animal model, and we are lucky enough to have CTNS deficient mice. And as Corinne said yesterday, there is a lot of similar symptoms, and we accumulate cystine. As donor of stem cells, we used the GFP transgenic mice. So these mice are green. They are really green. And so they also express a CTNS gene. So they have a functional gene in their cells, but also a green reporter gene that will allow us to follow the cells after transplantation. So we take the stem cells from their bone marrow. We have to irradiate the mice before the transplantation, and this is true also for human. If we want the stem cell to engraft in their bone marrow, you have to make space. So to make space, in the mice we do irrigation, but in human you do chemotherapy. And this is what we saw. Sorry, Kevin, I didn't put the negative control, because in the negative control you don't have any green cells, anywhere. But this is what I saw the first time when I went to the microscope, the confocal microscope, and I saw after 4 months post-transplantation only, I saw tons of green cells. So this means that, and this was in the kidney. So this means that the cells that I transplanted in their bone marrow migrated only in 4 months post-transplantation abundantly into the kidney. So that was a very good result, because this kind of abundant engraftment of bone marrow-derived cells in the kidney has never been published for any other disease. So this was the spleen. I mean, as you can see, it's all green. We also found some in the muscle, and we could see that they were like myocytes. So that could be also good news for the muscle weakness. You know, if they can go and replace the bad muscle cells. In the brain, you know, and you see here how they migrate. You know, this is the stem cells that migrate into a blood vessels. They go, and after they migrate into the tissue, and they go and replace the bad cells into the brain. And this is the eye, and this was done by Dr. Jester and Dr. Simpson. We saw also plenty of bone marrow-derived cells in the eye. So what about the cystine? So now we have the cells that migrate a lot into the tissue, but does that do anything on the cystine content in the tissue? So we measure the cystine content at 2 and 4 months post-transplantation in the different tissue. And so these are mice in gray. They are mice that received CTNS deficient hematopoietic stem cells. So they don't have a CTNS gene, and that was our control. And you can see here that these mice continue to accumulate cystine with time. So there is no effect. If you transplant stem cells that don't express a CTNS gene, you have no impact. But the mice that received stem cells that express a CTNS gene, you can see that the cystine drop, you know, and continue to decrease with time in every tissue. So that was a very good news, because not only the stem cells engraft abundantly into the tissue, but it lead to a significant cystine decrease in these tissues. So what about the kidney? Does that have any impact on the kidney function? Because now the next question is that, can we rescue the tissues? And also, if you notice, I made this study in very old mice, and as Dr. Max said yesterday, this is the age where they are in renal failure, and they are also almost dying at that age. So if we can rescue the kidney at that age, this means two things. Not only we can rescue the kidney, but also this means that the treatment is stable with the time. So for the whole life of a mice. And so you don't need to be expert in kidney to see that this is a really bad kidney. You know, this is a wild-type mice. This is a CTNS deficient mice. And at that age, the kidney is really in failure. So some of our treated mice will look like that. And as you can see, the kidney was pretty much preserved. There are some focal anomalies, but the kidney function was normal, as good as a wild-type mice. And this still was in the very old mice. So this means that with this treatment, we could rescue the kidney, so that we can lead to to tissue preservation. However, some mice that were treated also didn't have a good kidney. And so finally I realized that between these two groups the difference was the quantity of stem cell expressing a CTNS gene in their bone marrow. So these mice have more than 50% of their stem cells that express the CTNS gene, and this one have less. And why I'm saying that? This means that the quantity of stem cells that express a CTNS gene matters, and that might be important also for any clinical trial we want to do later on. We have to remember that there is a threshold that we have to reach to get an efficient treatment. Again, at 15, 17 months old, a kidney histology, and you see, I don't know if you see well, but there are plenty of cystine crystals, and this is a treated mouse. And there is almost very few or no cystine crystals are seen in the treated mouse. So it seems like the cells are not going randomly into the tissue. It seems like they really target the cells that contain the cystine crystals to lead to their resolution. So this is, you know, pretty good results in the mice, so we were really happy. And that's why this work led to the first stem cell clinical trial in cystinosis. And this trial is called Allogeneic hematopoietic stem cell transplantation, and this means that we are taking the stem cell from a donor, and in this case the donor is a sibling donor, so we want a brother or sister of a patient. The doctor taking care of this trial are Theodore Moore and Donald Cohn at UCLA, but this treatment, this trial is done only to test the efficacy and the toxicity of this kind of treatment in human. The patient should have a sibling donor with fully compatible, so 10 out of 10 HLA match. So Betty said what, this is explain what was, we want them to be the most compatible possible. They must be 18 or older, or they can be as young as 13 years old if they do not tolerate cysteamine. So because of this strict inclusion criteria, all the patients tested were not fully matched. So we didn't find a fully matched patient, with his sibling donor. So we didn't, but there are still people getting tested that we know of. So, we might have a patient. But stay with me because it's not a big deal if we don't have a patient for this trial. So I just want to explain a little bit better what is allogenic. So you have the patient with cystinosis here, and you have a sibling donor that is healthy. So we'll take the stem cells from this person, so from a blood draw, we draw their cells and take their stem cells. This patient will go to a round of chemotherapy to remove their stem cells, and after we put the stem cell back to the patient. So the advantage of this allogeneic transplant is that you have 100% of the cells that will express the CTNS gene. So after this procedure, the patient will have in his bone marrow almost 100% of their cells expressing CTNS. And I said that it might be important for the efficacy of the treatment. But the problem about this treatment is that you have high risk of infection, and you have also, as Betty said yesterday, a risk that the host cells reject the donor cells. And after you start a chronic disease, so it's very risky procedure. So that's why it's not the treatment we want to develop for all the patients with cystinosis. This is just a proof of concept. What we want to develop for all the patients with cystinosis, if it's working, is what we call the autologous hematopoietic stem cells. So in this case, we take the stem cells from the patient goes through chemotherapy. In the meantime, in the culture plate, so in the laboratory, the stem cells are modified genetically using a virus vector that is been modified, but so it's not a toxic virus, but it can go to your cell and integrate into the genome and now express a functional gene. And after, the modified cells are reinfused into the patient. And this method has the advantage that it's much safer. Because you are using the whole patient stem cells, there is minimal risk of rejection, almost none. So the only risk you have here is if a gene goes to a bad gene when it integrates into the genome. If let's say it goes to a cancer gene. This already happened for some vector, and in this case, you can have, develop cancer. But the risk for that is really low, and there are a lot of studies, and that's why I will tell you what I have to do now to prove that this is a safe vector. The disadvantage of this also of this strategy is that when you modify genetically ex vivo the cells, not all the cells will express a CTNS gene. So we are hoping that we'll be able to reach the right threshold with this method to have an efficient treatment. So just to summarize what I just said, this is, let's say, the hematopoietic stem cells from the patient. So you see that the CTNS gene is non-functional, so you have no cystinosis, and you don't see, but there is no cystine, the cystine stays stuck in the lysosome. If you use the virus and what we call, it's our lentivirus vector, that we call PCCL CTNS, so this is the human CTNS gene that is subcloned into this virus. And why we are using the human gene? Because this is the, if this is successful and safe, this is the same construct, the same vector we'll use in human. So it's already ready. So this is our antivirus vector. We add that in the plate. That vector enters the cells, goes into the DNA of the patient, and integrate the new CTNS gene. Now you have a new protein, functional, and the cystine should be able to go out the lysosome. So, as always, when you want to do something, you have to prove it in the animal model. So we are using our CTNS mice again. And we took their stem cells, we put in the plate. We are using our lentivirus vector, expressing a human gene, but it's fine. We put them in the plate, and we transplant them in a CTNS deficient mice after irradiation. And what we have done is that we have done a lot of things, but I will just summarize in one slide. We checked the male 8 months post-transplantation for the impact of this treatment, and we checked for this kidney. So this is the cystine content in the mice. As you can see, so these are the non-treated mice, and these are the treated mice with CTNS, so with our lentivirus vector. So you have a significant decrease of the cystine in the kidney. The kidney function is improved, and you have no more cystine crystals in the kidney. So even with this method, we could reach a great success in the mice. But again, in human, we will. I mean, with mice we can put a lot of virus, and so we still have to make sure we can reach the same kind of threshold of cells expressing CTNS in human, and we'll see if it can work. So where we are now, because now we have done the proof of concept for the wild types hematopoietic stem cells. We showed that we can use also the autologous, so we can use the patient stem cells, modify them with an antivirus. So we would be ready to go, right? But it's not like that, because this is called like a new drug. This is because we are using a lentivirus vector. It's considered like putting a new drug on the market, like RP103. So we have to go through the whole FDA process. And so our new drug will be the PCCL-CTNS gene. This is our new drug we want to put on the market. And, I mean, not on the market, it won't be for sale, but we want to develop it. So, we had to go through what we called an investigational new drug. And we did last April, so I think, I don't know if you remember, but I told you that I had my first contact with FDA last April, so 1 year ago. And I had to file a big file already to say, okay, this is what we call a pre-IND. It's a first screening, say yes or no, just forget it, don't go, or maybe you can submit a pre-IND. So they told me at that time, yes, you can submit a pre-IND. And we submitted, and thank you to Betty, because now Betty Cabrera is the study coordinator of a future clinical trial for cystinosis. And so she and I put together a pre-IND file, and we submitted last March. And at the beginning of this month, on April 4th, we had our teleconference, and they approved everything we have said, and they were kind of enthusiastic. We had good review, and everything we proposed was good. So they said, yes, you can go forward. And by the way, I take this opportunity to say, the registration is really important, because we will need a lot of data from the CCIR now, because one of the jury, and there were 14 members, one of them still said, yeah, but with cysteamine, it's kind of, they are pretty much healthy now. And we're like, no. so, I mean, fortunately it was only 1 out of 14, but we still hear it. So we will need data to prove that this is not true. And that's why the registration has been made at the beginning, and it's a tremendous source of information. So now that we approved our pre-IND and what we want to do, now what we have to do is the pharmacology, toxicology studies of our new drug. And now the problem starts, because now we have to do a lot of studies that will be closely monitored by the FDA. We have to give them, a report all the time in the right format, and this will take 2 years at least. So what we are going to do is just quickly, we have to use a preparation of virus that will be what we call good manufacturing practice comparable. So this means that it's not done by me anymore, by our lab. It's done by another source who can make the same virus that will be used in human. So it's very expensive, because now we have to use the right virus preparation done by this company. And because of the extra money I had from the Cystinosis Research Foundation, I didn't have to wait to get funding, but it's already ordered and almost done. So we will be able, once it's done, we can start. So we saved at least 5 months here. So now we are almost at the point, because they gave us, we already titled the final preparation. So they will send it, now it needs to get certified, and we will get it in a month or so. So what we will need also, we need blood from healthy donor, but also from cystinotic patient. And we need, I think, at least 3 volunteers, because we need to now optimize and verify the toxicity in the human CD44 cells, so the human hematopoietic stem cells. So we have IRB approved, so this means that we just got approved, and also again thank you to Betty for that, at UCSD, so we'll be able to get the blood at UCSD. Ranjan Dohil and Nadine Benador will be the doctor who, if they see patients, they will be able to draw blood for us. And Corinne Antignac will identify the CTNS mutation, because we really need to know what kind of mutation we are dealing with. And this will be very important for the next step of the study, how to control the efficacy, the impact, and everything. We need to know the mutation. So the volunteer will have to have a mutation screen, and later on for the clinical trial, anyway, we'll have to have all the mutation screen. So the first step will be the transduction optimization. So we will put the stem cells in the plate and try plenty of density, plenty of concentration of virus, to see what is the best optimal concentration for having a good transduction of the cells. And we have to stay in a right range, because the FDA say you have to be in a 1 to 3 range, but anyway, we have to, so it's a lot of optimization to make sure we have that, so we'll do that first. And after we have just in vitro, different in vitro assay, using the human cells, showing that once they have a virus in their cells, they don't become crazy. They are just regular stem cells. So we have to prove that by different assays. We have also a series of in vivo studies, and they are called the serial transplantation. And here again, we are using the mouse model. We will take the stem cells, put our virus, but in this case, the GMPC preparation. We put them in a first group of mice, and we are dealing with a lot of mice in this case. Again, we want a good group of mice. And we take the stem cells from this group to put in another group. And we want that because if there is anything wrong with the stem cells, this puts so much pressure on the stem cell that you will see it. And from these 2 groups of mice, we have to do tons of studies that will cost tons of money and tons of time. To summarize quickly the situation. But it needs to be done, and it needs to be done right. So, because again, the FDA is really tough. So, so this is what we will be able to do. And as I said, as we were able to buy the GMPC preparation, in 1 month or so, we should be able to get it once it's certified. And we will be able to start all this. You know, we have already, you know, tried to put together everything to not waste time. So we are doing our best to go the fastest we can. And so, and this was possible because of the funding of the CRF. And so, so this is where we are. And so it will take at least 2 years, I have to say. So, and so if we do that in 2 to 3 years, what we hope is to be able to write an IND. So this is the final IND, where that RP103, you know, you have probably heard all these words. In the meantime, during that time, as soon as the funding starts and we know we are good to go, we'll have all the panel of doctors, you know, that will work for 2 years to plan the clinical trial, and of course Dr. Dohill, Dr. Troner, I mean, all the doctors of UCSD will be there, and all the, you know, we need a doctor for each specialty, you know, the brain, the eye, the kidney. So we will make sure with Betty to have all this organized, to be ready in 2 years. If we submit our IND and if everything goes fine, if there is no adverse event, in this case, they should approve us for the first clinical trial. So I just want to finish by acknowledging my group, Céline Roca, Sarah Ure, Betty Cabrera, Jay Sharma, Swati, and Athena, who is not in this picture, and Frank and Brian before. Person for sitting content, Corinne Antignac, and Marika Gubler and Vincer, and Dan Salomon. Of course, again, thank you to the Cystinosis Research Foundation for the funding. I got, since this foundation, since this funding, I got 2 NIH grants, because now they realize this is really a possible new treatment for multiple other diseases that we didn't think about, such like, because before when we were thinking about degenerative diseases like cystinosis, where all the tissues are tested, are injured, we didn't think about using hematopoietic stem cells. So this is a big turn in also the field of regenerative medicine, and so that's why now the NIH is really interested by this work and now is funding this kind of work. So, and thank you Nancy, thank you Jeff, and thank you all the family for your support.